Thanks, Matt. It's a pleasure to be here and talk about nutrition and the gut-liver axis and hepatotoxicity. And this talk is really designed to kind of lay the groundwork for several other talks that are going to come later. And I also, as Associate Vice President for Translational Research and Health Affairs, wearing another hat, want to welcome everybody to Louisville. And you'll be at the racetracks tonight. And I don't have any disclosures related to this talk. So in the next 20 to 25 minutes, I'm going to give a brief overview of gut barrier, obesity, and liver injury, talk about gut barrier and high-fat or high fructose diets, and NASH, which Matt gave a little introduction to, talk about gut barrier, dietary fat, and alcoholic liver disease, then some nutritional interventions and conclusions. So first, for the gut barrier, it's incredibly complex. And so as seen here, that was bad. Yeah, no. I'm going to not try that. So in green is the mucus layer, which is incredibly important and not well understood. The little blue things up at the top are your gut bacteria, and people tend to focus on that. The purple cells are the intestinal epithelial cells, and the red tight junctions are critical for preventing bacterial toxins from translocating across. We also have Panna cells, which are critical immune cells, and again in the blue, goblet cells, which secrete mucin. And so a screw up in any one of these things can cause gut barrier dysfunction and ultimately liver problems and metabolic endotoxemia. Now, I'm a gastroenterologist, and classically gastroenterologists, when they think about the GI tract, they think about the stomach secreting acid, the small bowel absorbing nutrients, the colon fine regulating water. But if you look, there's also a gradient of bacteria going down. And actually, there are many more bacteria in our GI tract than there are cells in the body, many more genes in our GI tract and the bacteria than there are genes in the human body. So really, we're just a receptacle for our gut bacteria. And as shown here, the surfaces of the body have many, many different types of bacterial cells residing there. So if you see here, classic phyla in stool are the Bacteroides and Firmicutes, which is very, very different from over here just in the mouth and the tonsils, and highly different here in the vagina. And we measure bacteria classically by 16S ribosomal RNA. We're interested in bacteria really for what they produce. And so that's what our group is focused on more, is not what bacteria are there, but what are they making. And so that's really the metabolomics part, and we're going on automatic pilot here for a second. So I've shown this slide just to show what a short course of antibiotics can do to your GI tract and the metabolites that are produced by gut bacteria. And so it causes a decrease in short-chain fatty acids, branched-chain amino acids, and altered bile acids. And this is highly relevant when we come back to what we see in alcoholic liver disease. Now, this is all important because, as Matt said, we're an overweight country. 2/3 of Americans are overweight. A third of us have NAFL, and this is really the major health problem in the United States and emerging in the world. And many people feel that metabolic endotoxemia or metabolic infections are really the driver for this process. And so on the left here is our cartoon of this concept. So altered gut flora and increased gut permeability can lead to increased translocation of endotoxin and gut bacteria, increased TLR4 activation, TNF production, liver injury, and systemic inflammation. On the right is the cartoon by Canny and co-workers who have also popularized this concept that basically metabolic endotoxemia can cause all of the complications of the metabolic syndrome, including NAFL. So we look at this process through many different ways. One is starting out looking at high fat and high fructose diets leading to NAFL. And again, Kentucky is a leader in this. We have many food chains such as Papa John's, KFC's headquartered here. In our dietary models that we use, we classically use the high fat or high fructose diets. Interest in this was stimulated by groups from Washington University in this important trial where they showed that germ-free animals had, in white, had less body fat, yet they ate more and had less oxygen consumption. So if you had a pill that could reproduce this, you would be a bazillionaire. And this is compared to conventional animals or germ-free animals that were then given normal gut bacteria in black. It was then shown that genetically obese mice, either the DB/DB or OB/OBs, had increased gut permeability as assessed by horseradish peroxidase or by serum endotoxin levels. They had endotoxemia. More importantly, if you just feed animals a high-fat diet, as shown here, at 1 week, you can detect bacterial DNA in their bloodstream. And very importantly, it goes to the mesenteric adipose tissue before it goes to mesenteric lymph nodes. And so the adipose tissue isn't just a benign glob of fat there. It's very biologically active and actually takes up gut-derived toxins. Another pivotal study from Washington University looked at the OB/OB mice and showed that they had altered gut flora with an increase in firmicutes and a decrease in Bacteroides. They then transplanted the gut bacteria from these obese and lean mice into germ-free mice. And the germ-free mice that got the gut bacteria from obese animals ended up with more body fat. Now, an even more important study from my perspective, and much less well known, is this article in Gut where they did a study that we do all the time. We feed animals a high-fat diet. And not all the rats or mice respond the same way. So you have some over here that don't gain much weight. Most of them gain a fair amount, and then there are a few of them that become really obese. And so what they did is they took the stool from the non-responders, the ones that didn't gain much weight, and stool from the rapid responders, and transplanted that into germ-free mice, then fed them a high-fat diet for 16 weeks. And the ones that were the rapid responders in black had much worse insulin resistance, had more fat in the liver as shown here, and much more fat histologically as shown up here. So this shows that the phenotype can basically be transferred through a stool transplant. Now, You don't have to have animal models. We have a classic human model. A movie was made out of this, Super Size Me. So one of our fellows at the time, who's now on faculty at the Mayo Clinic, actually froze the frames in this movie while they were looking at this guy's medical record. And so you can actually see why they stopped him from continuing to go to McDonald's and eating. So his liver enzymes here over time were just going up dramatically, and so he had incredibly severe NASH due to this. So we've talked about high-fat diet. What about high fructose diet, which has been one of our research interests? And so this was started by Ina Bergham when she was a postdoctoral fellow here, actually working in Gavin Artiel's lab. Miriam Voss is going to talk next, and Ming Song from our group. And so what we initially did was feed mice either just regular diet. Everybody gets the same diet, but they either get regular water, artificial sweetener, glucose, sucrose, or fructose. And when we sacrifice the animals, you can see the glucose and sucrose animals have a lot of visceral adiposity. The animals that got the fructose have less visceral adiposity, but more fat in their liver. And that's highlighted here by much higher hepatic triglycerides noted in the yellow, higher 4-hydroxynonenal, and greater endotoxemia and much greater hepatic TNF. So what's the gut link here? We've shown that these animals have alterations in their tight junction. You saw the endotoxemia. And importantly, when you alter the diet and give them antibiotics, so you're altering the gut flora, why you totally protect against the endotoxemia and the fatty liver. So somehow the gut flora is playing a role in the fatty liver. Ina recently has shown that all the tolls are upregulated in the liver with fructose feeding. And again, when you give antibiotics, you protect against this. Now, what if you have two dietary alterations intersecting? And this clearly happens. So Ming Song from our group looked at marginal copper deficiency, which is a problem in our diet, and high fructose consumption. And the marginal copper deficiency and fructose over here cause much greater fatty liver. The fructose causes a lot. The marginal copper really doesn't do hardly anything. And if you look at liver enzymes, it's this group, the combination, that gets elevated liver enzymes, and they have much higher hepatic iron. So I see several patients a month that have this problem in my clinic. So they come in, they have a low copper and ceruloplasm, so they're sent in because their physician thinks they have Wilson's disease. Or they're sent in because they have high iron levels, so they think they have hemochromatosis. And instead what they're doing is drinking 12 cans of regular Mountain Dew a day. And so we try and either get them to drink Diet Pop or get over to water. And if they do that, this all goes away. So something that's very, very fixable. Now, and Miriam will tell you when you go to nutrition meetings, there's this huge battle over is it fructose or is it just any type of sugar that's bad. And so there's been incredibly interesting articles looking at the endogenous fructose production. And they've knocked out two enzymes that are responsible for endogenous glucose conversion to fructose in these studies. And so here are wild-type animals fed a high glucose diet for 16 weeks. They get fatty liver, the knockout stone. Here's the histology, fatty liver, knockout stone. Liver enzymes, alterations in ALT and AST in the wild type, but not the knockouts. And This particular enzyme here, aldose reductase, has been of great interest to our groups here at Louisville. And so maybe it's just sugar drinks in general that are bad, and Miriam is going to talk about that in our next presentation. So in general, high fructose actually can cause all aspects of the metabolic syndrome. Now, in a parallel fashion, we see that alcohol can cause alterations in gut barrier function, endotoxemia, activation of tolls, TNF production, and inflammation. And dietary fat can markedly modulate that, and this work was done by Irina Kirpik. Now, it's been long recognized that there's altered gut permeability in alcoholic liver disease. This is human studies looking at a marker of gut permeability, and just alcoholics coming into a detox unit, as they come off alcohol, they get better. And there's been historical data suggesting that dietary fat plays a role. And so there's both human studies and older studies from Amananji suggesting that unsaturated fat, and especially N6 fatty acids, are required for the development of alcohol-induced liver injury. So Irina fed animals an alcohol diet either high in unsaturated or saturated fat. And the unsaturated is in the orange here, and you can see unsaturated plus alcohol causes a decrease in all of the tight junctions that we measured. So it impairs tight junction function, and you get endotoxemia. You don't get that when they're fed a saturated fat diet. The unsaturated fat diet caused an upregulation of all the tolls in the liver, just like Ina saw with fructose. And here's TLR4, and then ultimately hepatic TNF is markedly increased, but not in the saturated fat diet. Ultimately, this leads to a severe fatty liver in the unsaturated fat, but not the saturated fat, again with more liver triglycerides and an increase in ALT levels. And if you look at what happens to the gut flora, you can see that similar to humans, the two major phyla are the Bacteroides and the Firmicutes, so in the brown and green. And this is very consistent over time in the saturated fat diet with alcohol. But you can see in the unsaturated fat with alcohol that ultimately by 8 weeks, there's an increase in new bacteria here. So new phyla coming in, actinobacteria in red and proteobacteria in blue. And this is totally blocked by the saturated fat diet. And we see this with several of our other interventions in alcoholic liver disease that I'll talk about later. So what are some of the mechanisms for this? Well, linoleic acid can get metabolized over the lipoxygenase pathway to oxidize products of linoleic acid, or oxalams, that are highly inflammatory, and they're also ligands for TRPV1, which activates the inflammasome and causes an increase in IL-1 and IL-18 that we see in our patients with alcoholic liver disease. And shown here, we're looking at barrier function in KCO2 cells first with linoleic acid, with or without alcohol, and our different oxidized products of linoleic acid. And you can see here the 13 HODIs plus alcohol basically just totally knocks out barrier function. They also markedly alter mitochondrial function, and so we think these are major mechanistic pathways. So in summary, linoleic acid exacerbates alcohol-related hepatotoxicity. Saturated fat protects. So this is kind of teleologically doesn't seem right. This is different than what you've heard in other types of liver disease. and ox lambs may be partially responsible for the gut barrier dysfunction and the liver injury. In the last few minutes, I'm going to then talk about 3 nutritional interventions. So can we use nutrients to treat or prevent liver disease? So we'll talk about a trace metal zinc, a fat butyrate, and a probiotic lactobacillus GG. So first for zinc, these studies were done by Matt Cave, Dr. Zoe, who's now in North Carolina, and Bert Watson. And this is a zinc deficient patient. So this is one of the first zinc deficient patients that was described by our group back in the 1970s with classic skin lesions around the eyes, nose, and mouth. This is an alcoholic cirrhotic. Zinc has many critical functions. We're going to focus on zinc finger function today. And in alcoholic liver disease, there are major alterations in zinc metabolism. So you have decreased intake, poor absorption, and increased excretion. Unfortunately, I wasn't smart enough to figure out what was really happening, though, with the altered zinc metabolism. It was Dr. Zoe that did this when he showed that if you have oxidative stress, you lose the zinc from the zinc finger proteins and lose transcriptional activity. And so this is a critically important function here. So you've now lost your DNA binding activity. And that's shown here in an animal study. So we're looking at HNF4 alpha, and again, this is binding activity. So your protein level's fine, but it's your activity that's abnormal. So here's alcohol, alcohol plus zinc. If we give these animals that are fed alcohol zinc in their water supply, here's just the alcohol alone, alcohol with zinc. Markedly protects. The next thing we'll talk about, we've gone from a trace metal to a fat. And these are some of our metabolomic studies. So we feed these animals a high-fat diet for 6 to 8 weeks, or we can feed them an alcohol diet and then go take their stool and do metabolomics. We can do metabolomics in the liver or the serum too, but we're focusing on stool. And this is the alcohol study. So in alcohol, you see a consistent picture with a decrease in short-chain fatty acids, altered bile acids, and a decrease in branched-chain amino acids. So almost identical to what we saw with the antibiotic feeding. So we focused on short-chain fatty acids, and specifically butyrate, which is critical for, it acts as an HDAC inhibitor, and it can act through G protein coupled receptors. But we were focusing on HDACs, which I don't have time to show you now, but what I will show you is what happens when you give butyrate. Can you take a short-chain fatty acid that's decreased in the stool, give it orally, and protect against the liver disease? And so we gave tributrin, which is a stable prodrug of butyrate, and here you see the endotoxemia with alcohol, protected with the butrin, increased fatty liver, protected. And it also protects against the liver injury and inflammation. The last nutritional intervention that I'll show you is probiotics, and specific lactobacillus GG. And these studies were done by Winky Feng, Shirish Barve, and our collaborators at Baylor. And this is a specific probiotic, so different bacteria do different things. We use lactobacillus GG actually because of its ability to regulate HIF and intestinal trefoil factor, which is important for mucus stability in the intestine. The study design here is very important. So this mimics clinical disease. So we fed animals alcohol in their diet for 6 weeks, We then continued the alcohol after they developed liver disease, but intervened with the LGG. And I would also say that Dr. Feng has now gone on to show that you don't need to use the live bacteria. You can just use the supernatant that the bacteria is growing in. So the bacteria are actually producing products that are critically protective. And if you look at endotoxemia, up with alcohol, down with LGG. If you take mesenteric adipose tissue, so we gavage these animals before they're sacrificed with GFP E. coli, take out their mesenteric adipose tissue, and here are the regular animals. Here are the animals that are treated with LGG. So basically no viable bacteria. If you look at TNF, up with alcohol, down with LGG, MPO, up with alcohol, down with LGG, liver enzymes are improved, and most importantly, here's the histology with alcohol, improved with LGG. And again, I don't have time to show you, but we've published an article documenting that as time goes on, the animals develop abnormal gut flora, just as I showed you with the high-fat diet. And if you intervene at 6 weeks with LGG, why you can correct the alterations in gut flora as the animals improve their liver enzymes. So in conclusion, there's extensive crosstalk between food, alcohol, toxins, which I haven't talked about, but other people will later today, and the gut microbiome and the metabolites that are made by the gut flora. Gut barrier dysfunction plays a critical role in fatty liver and steatohepatitis. The gut barrier provides multiple targets for prevention or treatment of liver disease. However, this is likely to be a very possibly disease-specific or even individual-specific issue, so this isn't going to be a one-size-fits-all solution. Thank you.
We have time for questions from Dr. McClain. Well, I had one, actually. The ALT increases that you see in some of your mouse models, like the one you just talked about, are really pretty small. Do you
Right.
actually see histologic evidence of necrosis in those models?
Yes, so the liver enzyme abnormalities that you see with the chronic feeding models are not particularly impressive. Actually, the liver enzyme abnormalities that you see in alcoholic hepatitis aren't particularly impressive either. So that's one of the interesting things about alcoholic hepatitis. You can have really bad liver disease, but modest elevations of ALT. So you do see some necrosis. With this model, you really never see significant fibrosis. If you want to induce more necrosis, there's the what's called NIAAA model, where you feed alcohol for 10 days, and then you give a big gavage of alcohol on the 10th day, and you see much more necrosis there. So that simulates alcoholic hepatitis.
So talk
Dean.
about the zinc effect.
Right.
What does zinc do to the microbiome?
Yes, so the question is, what does zinc do to the gut flora? And actually, nobody studied that. That's a great question. And actually, a question a reviewer asked on one of my grants. Hope it wasn't you, Dean.
No, but you studied it.
And what I told the reviewer was that would be great, but it's actually expensive.
Tim?
How are you measuring gut barrier dysfunction? What's the functional assay that you're using to look at that?
Yeah, so that's what I was, when you look at gut barrier dysfunction, you can either look at the gut flora, you can measure endotoxemia, the bacteria going across. We measure tight junctions, so there are about 20 tight junction proteins that you can look at. So there are a host of different things that you can do. Then you can look at markers of things that go across the gut barrier and measure those. So you can measure proteins, you can measure gut-derived toxins, and actually we do most of them. Yeah.
So there are so many different rodent diets available for studying aspects of obesity and NASH, and perhaps suggest one to the group that you feel, recapitulates.
Yeah, so the question is, what rodent diet should you use? And that's probably an incredibly important, well, it is the important question. And I'm not sure that there's a right answer for that. And as I showed you, alcohol and NASH are very, very different. So it's probably more saturated fat that's important in NASH, we think. In alcohol, it's unsaturated fat. It's not only unsaturated fat, it's specifically linoleic acid. And so the important thing is when you read scientific papers is knowing exactly what's in the diet, which isn't there a good bit of the time. And so when people talk about high-fat diet, it just drives me crazy. And so, one thing for reviewing papers, reviewing grants, you have to know what's in the diet.